Okay, so I'd like to start a special lecture. Guests, friends and colleagues here. This is a greatest honor for me to introduce Dr. Nicholas Lydon. Dr. Lydon was born in Scotland and completed a PhD in biochemistry at the University of Dundee in Scotland. In 1985, he became the leader of the protein kinase inhibitor program at Ciba-Geigy, currently Novartis. Together with Dr. Brian Drucker in academia, Dr. Lydon and Dr. Drucker discovered Gleevec, imatinib. Before this discovery, nobody believed that the selective inhibition of a single molecule has therapeutic effect against cancer. He has received many outstanding awards, for instance, the Lasker Awards in 2009, JapanPrize in 2012, and the Fellow of the Royal Society in last year. I sent my enthusiastic invitation mail to Dr. Lydon with Damemoto, like this, Damemoto. I received the first mail from him in July and last year. The answer was, oh, yes, he accepted. So I jumped with extraordinary excitement. Dr. Lydon, are you ready?
Thank you very much for the kind introduction. And what I've been asked to do is, for the first part of my talk, I'm going to give you a bit of history on imatinib. And many of you may be familiar with the story, so bear with me. And in the middle, I'm going to give some anecdotes which are probably not obvious from the history that's been written, but most drug development efforts have problems you run into, and I thought it would be informative to show sometimes how difficult it is to get things into clinic and then through clinic. And then the second part of my talk, I'm going to give you a flavor of the work we're doing on the next generation of very selective kinase inhibitors that we are doing at Blueprint Medicines in Cambridge. So this gives the rough outline of the talk. Firstly, I'll describe CML. I think a lot of you are familiar with this disease, the discovery of imatinib very briefly, and some of the examples I'll show you in the development of this, which were probably early examples of the use of biomarkers and surrogate markers in clinical development. Problems along the way, and then, as I said, what we're doing at the moment. So when I started in the field of protein kinases, I was a naive biochemist at the University of Dundee. I was working on adenylate cyclase and the activation of the cyclic AMP-dependent protein kinase. And it wasn't a very good project, and I was struggling, but next door to me was the lab of Sir Philip Cohen, who was one of the pioneers in reversible phosphorylation. And he was studying the regulation of glycogen metabolism and its regulation by a cascade of protein kinases. And I was really impressed by this a very simple system that reversible phosphorylation could control a very complex metabolic pathway such as the regulation of blood glucose through this process of reversible phosphorylation. And it reminded me of the sort of wiring diagram in this case, my car at that present time. It reminded me of the switches and relays that are involved in regulating the mechanical features of that car. Now, unfortunately, this was an British car, and these relays and switches rarely worked. So I would say it was probably an example of a diabetic vehicle. So when I finished my PhD, the field of retroviral oncogenes was evolving. And in 1984, I read this review that was in Scientific American, written by Tony Hunter. And this really struck me because it made a connection between the retroviral oncogenes shown here, which were characterized by all containing a kinase domain, and the ability of these oncogenes, when they were deregulated, the kinase activity was deregulated to cause a number of cancers in animal systems. So shown here, I think you're all familiar, in this case, the Maloney-Murine leukemia virus. It's able to pick up, transduce a gene, in this case a protein kinase. These protein kinases are modified usually by removal of regulatory sequence and attachment of the structural gag protein. Normally these have high kinase activity relative to their normal cellular counterparts. So this is the archetypal protein kinase SARC, and at that time really none of these had been implicated in human disease with the exception of one, which was the Abelson protein kinase. And shown here is the viral homolog gag being substituted for the N-terminal regulatory part of the kinase, and this is an acutely transforming retrovirus. So why that was important was the work of Nolan Hungerford, followed by Janet Rowley, had identified the Philadelphia chromosome. And this was the first cancer to be linked to a genetic abnormality. So CML, the discovery of the Philadelphia chromosome, and then Janet was able to show by the technique of high-resolution banding at the time that this was in fact a translocation between chromosome 9 and 22, resulting in this abnormal Philadelphia chromosome. And this can be seen shown here in metaphase spreads, and also subsequent techniques such as FISH, and more recently by PCR or deep sequencing. So what is the molecular consequence of this translocation? So shown here is chromosome 22 and chromosome 9. There are a number of breakpoints in either the BCR gene or the Abelson gene. They result in two main mRNAs, which result in two proteins. One is the P190 BCR Abelson, which is characteristic of ALL, Philadelphia positive ALL, and then two forms of P210 BCR Abelson. And shown at the bottom here is what happens to the protein. Here is the C. Abelson protein with the regulatory domain and the catalytic domain. This is the V. Abelson protein, which I've previously shown you, where the N-terminus is substituted by a GAG protein, and this is the BCR Abelson protein with this substitution of the N-terminus. So the consequence of this substitution in both of these, in the retrovirus and the human protein, is elevated protein kinase activity due to removal of regulatory constraints. So what does this protein do? So the cellular consequence of this translocation is there is a mutation in a early stem cell, bone marrow stem cell. This gradually, at the early stage of disease, cells can differentiate through to their functional effector cells. As the disease progresses, there is an accumulation of increase in white blood cell count and the number of immature blast cells in the circulation. And this shows the clinical course of this disease. Chronic phase, lasting 5 to 8 to 6 years, followed by what's known as accelerated disease, and subsequently blast crisis, which is basically an acute leukemia. And shown here is the blood picture, white blood cells, normal individuals, and showing in the chronic phase there's a large elevation of white cell count in the circulation and increasing numbers of circulating blasts as the disease progresses. So this slide summarizes the extensive work that was done by the chemists in our group after we identified what was an early hit molecule through to the final discovery of imatinib, which we were able to patent in around about 1992. So when we look at this molecule, this was basically a very weak inhibitor. It had activity against protein kinase C. It was ATP competitive. And doing structure-activity relationship studies, we were able to determine that this benzimide was extremely important for cellular and activity and potency. The attachment of this methyl group and in the ring system here was critical for converting this from a serine threonine kinase to a tyrosine kinase inhibitor. And it's thought that this is due to being able to rotate around this rotatable bond to move these ring systems out of plane. We didn't do a vast amount of analoging around this end of the molecule, but over here it was critical to add the N-methylpiperazine group to improve its pharmaceutical properties. Although this part of the molecule was active and showed almost the same profile as imatinib, this was a very badly behaved pharmaceutical compound, as it had low solubility and other issues. So shown here is the specificity of this. In our assays, and I must say at that time we'd struggled hard to get as many kinases as we could into our assay system. We had around about 50 protein kinases in our panel, and we were extremely proud of this as we thought it gave us a good read on selectivity. The compound inhibited Abelson, PDGF receptor, and KIT in these assays. And shown on the next slide is the data from back there. This is cell-based inhibition, which matched its biochemical profile, inhibition of Abelson, PDGF receptor, and KIT, with no effect on a number of other kinase pathways. So looking forward a few years, we were actually able then to look at the absolute selectivity of imatinib using the Discoverex affinity binding technology, which I'll describe a little bit later. And I think it's gratifying that the selectivity we found there is pretty similar to that we described earlier on. And I think that's obviously a matter of luck. But I think that selectivity is an important for, or one of the key things we're aiming for at the moment is get high selective drugs, because lack of selectivity we interpret as adding a lot of risk with respect to the side effect profile and off-target toxicities. So as you can see here, imatinib binds and inhibits the non-phosphorylated form of the enzyme with greater potency than the phosphorylated, the active form of the protein. And what we know now is this non-phosphorylated is an inactive conformation that has a preferential binding to imatinib. The phosphorylated form, the active form of the protein, phosphorylation on the activation loop stabilizes the active form of the enzyme, has a much lower affinity for binding imatinib. And the reason for this was revealed by the crystal structure of imatinib bound to Abelson that was solved by the lab of John Kurian. And shown here is the a little animation to describe the, the activation of protein kinases. So shown here is the inactive conformation. And the activation loop is shown in yellow. And an important part is the end terminus of this activation loop, which is known as the DFG. And in this inactive conformation, we refer to these as DFG out conformation. And the type of inhibitors that bind to this conformation are now known as type 2 kinase inhibitors. So in this animation, you'll see this is the active form going back to the inactive form. And there's a relatively large conformational change in the structure of these protein kinases when they go through this activation process. So what happens in its active form, going to its inactive form, there's a large cavity at underneath helix C, which is opened in this inactive conformation. And shown here is the structure of imatinib bound to the inactive conformation. Shown here in yellow is imatinib. In white is ATP modeled in the same position it would occupy in the active conformation. And as you can see from this, ATP is unable to bind to this conformation of the enzyme, whereas imatinib occupies this pocket in the back of the enzyme, which is only present in the inactive conformation. Why this is important is because really imatinib is not competing with ATP in this binding mode. Since ATP cannot bind, it's probably responsible for its very high potency in cells. Basically, it has a similar potency in cells as in biochemical enzymes, which you would never expect from a kinase inhibitors. So that was retrospective. That was after the effect, but it was extremely important work because it opened up the whole field to the synthesis of these highly selective type 2 binding mode inhibitors. So when we look at the cellular effect of imatinib, this was a collaboration with a number of groups, Carlo Gamcorti in Italy, and then the MD Anderson group, Bev and Talpaz, and collaborators. And what we did, we assembled a large panel of Philadelphia-positive leukemias, including CML and ALL. And as you can see, that all of these leukemias were highly sensitive to the killing effect of imatinib. Whereas if we took leukemias which lacked the Philadelphia chromosome, these were totally insensitive to the effects of imatinib. In a collaboration I started at that time with Brian Drucker, he had been working on a number of cellular models of CML, and these include in the 32D cell system. We also tried a number of other IL-3 dependent cell lines, Mo7E included, and shown here is a IL-3 dependent 32D which had been transformed to factor independence by introduction of P210 BCR-Abelson. And as you can see, 1 micromolar of the inhibitor completely blocked BCR-Abelson phosphorylation. It had no effect on the parental cell line in the presence of IL-3 as monitored by phosphorylation of JAK2 and its pathway. And over here, you can see the cell system, introduction of the BCR-Abelson gene, shown here, resulted in factor-independent growth. In the presence of imatinib, these cells were unable to grow. So the development of a drug takes requires many different skill sets, and I've sort of listed a lot of activities that had to occur to get imatinib into clinical development and onto the market. So I'm going to focus on a few of these, which I think will be interesting anecdotes that probably haven't been written about. So some of the translational medicine aspects, which were very novel in the development of imatinib. Some issues we had in actually getting this project into clinic, because it was not obvious to the company that Gleevec would make any money, and there was a lot of resistance not to develop this drug. And then some of the aspects of clinical development, which nearly led to its discontinuation. So the translational medicine aspect was a collaboration with Brian Drucker, and was really a first in the field of looking for biomarkers and surrogate markers as early endpoints for clinical proof of concept validation. And the first thing Brian was able to do was to look at ex vivo bone marrow cell cultures from both normal volunteers and CML patients. And incubating these with growth factors to look at the GM lineage, and then the conolid-forming and the burst-forming erythrocyte units assays. And what was very interesting to us, this is the healthy volunteers, so this is 100% colony formation. In the presence of imatinib, there was no effect on colony formation. However, from CML patients, there was a dramatic reduction in the number of colonies from 70 to 80%. When we picked these colonies and analyzed for the presence of the BCR Abelson transcript by RT-PCR, we were able to see that we were highly selecting for the outgrowth of normal hematopoietic progenitor cells. To remind you that almost 99% of the cells going into this assay were Philadelphia chromosome positive. So this was highly encouraging to us that we could select for normal progenitor cells. And from those same patients, we were able to look for biomarkers. In this case, using antiphosphatyrosine monitoring, we can see cells from these patients. When you added imatinib, you could block the phosphorylation of a downstream target of BCR Abelson in both of these patients at a concentration of around about 1 micromolar. So moving on to the clinical development, and I'm just going to pick out a few very important pieces of data that really convinced us that the proof of concept was being achieved. So this is a graph from our paper in New England Journal of Medicine reporting the phase 1 study. And as you can see here, when we reached a dose of around about 300 milligrams per day, which from the pharmacokinetic studies we did equated to roughly 1 micromolar trough levels of the drug, we saw this dramatic normalization of blood cell count, which was durable for relatively long periods of time. So this was extremely exciting. Normalization from over some patients up to 100,000 cells per cubic millimeter, down to a normal in the 2,000 to 5,000 range. So dramatic normalization of blood cell count. And what was also incredible is this happened in 53 or 54 patients. And of those responding patients, we saw cytogenetic responses in 29 of these in very early clinical trials. 7 of these patients had a complete cytogenetic response, meaning we could not detect by cytogenetics the presence of the Philadelphia chromosome in any of these patients. And of course, we speculated that the potential for development of anti-cancer drugs based on specific molecular abnormalities could be treated by this type of approach. So looking forward, these are the results of the IRIS long-term study, imatinib study, which has been running for over 10 years. The data has been presented up to 7 years. So shown here is in red is the mortality, and blue is the incidence of CML prior to the introduction of imatinib in 2001. So you can see the incidence around about 4,000 a year, and of those, the mortality just north of 2,000 a year. After the introduction, there was a dramatic reduction in the mortality rate, and shown here by 2007 and 2009, there was a dramatic increase of prevalence. So the implications of this reduction in mortality is more and more patients were living with chronic disease. Shown here is the, up to 2011, is the mortality rate, and a significant subset of these are due to the emergence of acquired resistance to imatinib, and also in this are the acquired resistance to second-line therapy. So as you all know, this is a slide from the Sawyer lab at that time, early data 2002. This is a panel of mutations that had been identified, acquired mutation, resistance mutations. There are now many more. But what is important here is that imatinib is unable to inhibit any of these first-line acquired mutations. Two second-generation inhibitors, nilotinib, which is an analog of Gleevec with slightly higher potency, and dasatinib, a more broad-spectrum SRC inhibitor, aren't able to inhibit many of these acquired resistant mutations. The exception is the T315I mutation, which is resistant to any of the first and second line therapies. So fortunately, there is a third line drug, ponatinib, a very non-specific kinase inhibitor, which is able to inhibit this particular mutation. It has run into a lot of trouble due to its lack of specificity, a lot of cardiovascular events associated with therapy. However, for patients who have relapsed after second-line therapy, this is their only option. So there was a lot of pressure for the FDA to approve this for third-line salvage therapy. So, as I mentioned earlier, Gleevec is not a completely selective inhibitor. It hits two other kinases, the PDGF receptor and the c-KIT kinase, and this has led to its introduction in a number of diseases which have deregulations in these pathways. So for example, GIST, which has activating mutations in KIT and PDGF receptor. Systemic mastocytosis, which lack the KIT D816V mutation or have an activation of the PDGF receptor, are successfully treated with Gleevec. This is a very small percentage of the ASM population, and I highlighted this because I'm going to talk a little bit about a drug we're currently developing for systemic mastocytosis, a blueprint that is a mutation-specific inhibitor of the D816 mutant KIT. And shown here are a number of other diseases where Gleevec is shown to be effective. So this is the sort of timeline that's normally shown for the development of Gleevec, and you can see it looks very logical. Discovered the drug, went rapidly through clinic, and got registration. But unfortunately, that's not how things actually happen. So the first challenge was this. This is a marketing projection that was in our development proposal in '96 prior to going to clinic. And this is the estimate of peak sales by our marketing group for use in CML. And as you can see, they projected that the estimated sales for curative or highly effective therapy would be in the range of $60 to $140 million per year. And when you look at the development of pharmaceuticals, that is below the threshold where you would ever develop a drug, since the average development costs are north of 300 million at that time. So there was a lot of resistance not to move forward with this project. So along with my colleagues, we, because of the activity against KIT and PGF receptor, we were able to massage these numbers above 300 million to justify moving it into development. So this is the development plan, and we were able, based on this, the translational medicine work we'd done with Brian Drucker, to justify that we would go into CML as a proof of concept indication and then rapidly turn into clinical trials in solid tumors. The objective, based on clinical input, was to develop an IV formulation of the drug, to do a phase 1 trial, a proof of concept, and hopefully a registration, and then try to develop the oral dosage form before moving into solid tumors. There were some key questions at that time that were not obvious in developing such a concept. And the most important one is read: will surrogate endpoints be acceptable for registration in chronic phase patients? And we had to resolve this with the FDA before initiating these trials, because we were intending to use cytogenetic response and hematological response as a surrogate of survival. So this brings up the key questions for registration trials. Can cytogenetic responses be used as the primary endpoint, or will we be forced to do survival, which would entail very long phase 3 studies, which would be very difficult to justify given the marketing projections that the company had. So this is what actually happened in the trial. Just before we initiated the trial, in fact, we'd shipped the materials to our trial sites. We put in a final tox study of an infusion tox in dogs to look at the effect of the actual dosage form. And surprisingly, we had a very dramatic phlebitis caused by precipitation of drug from the IV infusion formulation, which killed a number of animals. So we had to terminate the IV formulation, and in fact, this particular event nearly terminated the program. In the meantime, the long-term tox from dogs came in while we were waiting for the development of the oral dosage form, showing that we had in a single species liver toxicity, specifically bile duct fibrosis, that was not reversible on discontinuation of therapy. So to be cautious, we had to add a monkey study, a 3-month monkey study, to see if this was a species-specific event. Luckily it was, and this led to the start of the phase 1 trial 2 years late. However, because of the rapid results we were able to see in the phase 1-2 study, the dramatic hematological and cytogenetic responses, we were able to get conditional approval of the drug prior to when we predicted on our initial plan. In addition to that, we initiated the IRIS study and got approval for first-line therapy in 2002, along with the approval for GIST. So this really led to a paradigm shift in how I thought of cancer, and also many in the field, which was, if you can take a disease, understand its molecular pathogenesis, pathophysiology, and come up with highly selective targeting drugs, this would transform the way we treat cancer patients from the classical chemotherapy type approach, cytotoxic chemotherapy, to more well-tolerated targeted agents. And I think the importance of this I'll address in the next part of the presentation. So this slide tries to illustrate a problem we had as I developed the Gleevec project and other projects at Novartis targeting different kinases. When I was at the University of Dundee in the late 70s working on the cyclic AMP-dependent protein kinase, there were really only 3 or 4 kinases known. But as you all know, after the discovery of the Sark oncogene and the tyrosine kinases, there was a rapid expansion based on PCR homology cloning of members of the kinase family, and by late 1990s, the complete complement was known, which is 518 protein kinases in the genome. Some of these may be pseudokinases, and of the total 518, 90 of these were tyrosine kinases. So our challenge became really difficult of how do we get selective inhibitors, and it also made it impossible to look at the literature and know what was a selective kinase inhibitor, since most of us only profiled these against a very limited subset of kinases. So in the around about 2004, I started working with a group at Ambit Bioscience in the in San Diego area, along with Patrick Zorenka and Dan Treba. We came up with this new kinase screening platform, which is now commercialized by DiscoverX and is based on an affinity-based biophysical binding assay rather than a biochemical activity assay. And this has a lot of advantages, since you can do apples-to-apples comparison of binding KDs across the kinome in the absence of varying concentration of ATP. So basically, it's a bar-coded kinase. This kinase can bind to an ATP-type analog, which is immobilized on a bead. In the presence of an inhibitor, this is competed away from the bead. You can then wash and, using quantitative RT-PCR, deconvolute this. The advantage is this is highly multiplexable, and also has a very large dynamic range. So it gives us the ability to look at the whole kinome for selectivity in one assay system. And these are the assays we developed at the time, 450 distinct kinases, covering most of the major kinases of the kinome. So what can we do with this? There are a number of aspects of this. So the first thing we did was we looked at the absolute selectivity of a number of known kinase inhibitors, and they range from very non-selective. One means it would inhibit the whole kinome, every kinase, and zero obviously going towards these are the most selective. So we give them an S score, and we set that at a specific concentration. For our example, this is S score at 3 micromolar in the biochemical assay, and it's the number of hits versus the number of assays. So it gives you this S score, and so these are the most selective inhibitors. Shown here is lapatinib, which is on the market and is one of the more selective kinase inhibitors out there, and shown here is imatinib. The other thing that it can be used for is driving a medicinal chemistry project where you actually know upfront what the absolute specificity of your inhibitor is. So shown here is a project we did at Ambit, the discovery and development of AC220, which is a very selective FLT3-ITD mutant inhibitor for use in AML. This is currently entering phase 3 studies, and shown here is the selectivity of this inhibitor versus a number of other published inhibitors. Very selective, hitting only a few kinases at doses you would expect in clinical use. So following that, I started a company known as Blueprint Medicine, and the real driver of that was the intersection of targets which were coming out of genomics and the technology improvement, high throughput sequencing, next-gen sequencing, and other techniques which were beginning to identify new targets in oncology. And the concept was to come up with very selective kinase inhibitors. And shown here is an example. I mentioned the Abelson 315 inhibitor T315I, ponatinib. It's a very non-selective inhibitor, and that lack of selectivity leads to its very problematic side effect profile in clinic. When we go towards more selective inhibitors such as lapatinib, most of the toxicities you see in clinic are on-target mechanism of action-based toxicities. So what we're aiming for is very selective compounds which hit one or a handful of kinases that can be used clinically with very low side effect. And I've listed here some of the reasons we want this. The ability to dose high. As we know from our experience with kinase inhibitors, you really want to block completely 24/7 the target you're trying to modulate. And you can't do this if you have toxicities resulting from off-target effects by modulating other kinases. We want to be able, the next step, to combine these with limited overlapping toxicities with other drugs. And there are lots of new drugs coming through now targeting different pathways, orthogonal pathways, where we would like to use these very selective molecules in combination. And because you know really the target you're modulating, it enables you to do early clinical proof of concept, as we did in the development of Gleevec, knowing that you're modulating the target, the intended target of interest, and that your effects are due to that modulation. So this summarizes what we're trying to do at Blueprint. We have the genomic side, and I'm not going to describe in any detail the genomics approaches we're taking. And on the other side, we have the chemistry approach, which is the construction of a library of very selective kinase inhibitors. And what we're looking for is this overlap where we have compelling genomic drivers that are kinases, and we have compelling, very selective kinase inhibitors that address that kinase. So a few words on how we've built this library. So we have a lot of knowledge on the structural basis of binding. There are many crystal structures and many chemotypes out there. But what we wanted to do is something completely different. We wanted to build a library that was agnostic to the kinase that was targeted. We didn't mind what kinase we inhibited, but we wanted to inhibit as many as possible with selective kinase inhibitors. So we selected in an iterative fashion around about 30 to 40 different scaffolds. And these scaffolds all have to bind in the ATP binding site of the molecule, of the target kinase. But what's different, they vector the substitution pattern slightly differently into the major pockets found in these kinases. And by iterative decoration of these with drug-like substance properties, we were able to come up with extremely potent inhibitors of a number of kinases. And the important thing is we monitored the selectivity of these libraries as we made them against the whole kinome. So we know, as we're synthesizing them, what they hit, and we were able to tailor and modify the decoration of these scaffolds to get as many inhibitors as possible. So this shows the current status of that library build exercise. We've carefully constructed around about 6,000 compounds. These are novel structures. They're differentiated starting points and multiple scaffolds. We have an 80% coverage of the kinome, and 60% of the kinome is covered with greater than 3 scaffold classes, and 30% with greater than 6. Then we have what we call tool compounds. These are potent and selective compounds that work in biochemical assays and in cell-based assays, which we think are similar can be used for actual target validation and interrogating novel biology. I should add that probably half of the kinases in the kinome, are kinases with unknown function or biology. If you look in Medscape for one of 2 of these kinases, there are no references on any biological function. So we've called these kinases of unknown biology, and we've accumulated a set of around about 100 selective inhibitors that block these kinases. And What we now need to do is try and work out a function for these. So this is a table from a publication from Stefan Knapp, who looked at what they referred to as tool compounds existing at the end. This was last year. And the criteria for these is they're highly selective and can be used for biological interrogation. And they classified around about 22 of these chemical tool inhibitors that they believed were selective enough to be used as tools. Based on the same criteria, we believe we have around about 200 of these tool compounds, and for many of these targets we have multiple scaffolds which address the same target, so multiple chemotypes inhibiting the same kinase. So what are we going to do with these? We can use these tools for a number of obvious biological experiments, identifying novel targets, interrogating new target biology, looking at combinations with existing drugs, and looking at undruggable targets. And shown over here are some of the approaches we're taking: sensitivity screens, phenotypic screens, synthetic lethality, resistance combination screens. And I'm going to just give one example of a collaboration we're doing with the Sanger Institute in Cambridge, where we are taking their panel of 1,000+ cell lines and testing 77 tool compounds that address kinases of unknown biology. And all these cell lines are genomically annotated, so the hypothesis is if we have activity against these cell lines, we are able to look at the genomic background that leads to this sensitivity. We have the data sets in hand at the moment, and we're in the difficult stage of interrogating them to see the genomic mutation context that leads to selectivity for these compounds. So what we tend to see is there are 3 classes or 4 classes of these inhibitors. There's a subset that has no effect on any cell line. There's a small subset that kills all cell lines. And then there are the 2 most interesting classes that probably 20 of them inhibit 1 to 10 cell lines, and the other one 10 to 30 cell lines. So I think those are probably the ones we'll focus on, but we have not got to the interrogation of the genomic profile that leads to this selectivity at the moment. So in addition to the tool compounds, we obviously want to develop some drugs, and I'm going to give you a flavor of that by illustrating one of these very selective compounds that we are progressing towards clinic. And this shows some of the projects we're working on. The first one I'm going to describe is a selective mutation-specific inhibitor of the D816 V mutation that's found in mastocytosis and a number of other cancers. We have a program in hepatocellular carcinoma targeting the FGFR4 receptor. This is due to the finding that the FGF19 growth factor, there's a focal amplicon that leads to high-level expression of FGF19 in a subset of hepatocellular carcinoma. And then these are 2 novel translocations that have been identified in our genomics program where we have selective molecules moving forward in chemistry. So looking at the most advanced program, this is, again, it's a mutation-specific inhibitor against the D816 mutation. It's present in exon 17 of the receptor. Almost all patients with systemic mastocytosis, 95% carry this identical mutation. In addition to systemic mastocytosis, this mutation is present in T821 AML. This is a bad prognosis mutation in this subgroup. And probably the largest class is acquired mutations, drug resistance mutation in GIST following second-line therapy. So if you look at the instance of mutations that are D816 as you go from first line to second line, there is accumulation of D816 mutations, which is the mutation which can't be treated by any of the drugs. So these are the three indications. This would obviously be the proof of concept studies we'd like to carry out in aggressive systemic mastocytosis. This shows the subtypes of mastocytosis going from the indolent disease, the aggressive systemic, through to the leukemic mast cell leukemia, which is rapidly fatal. So what does this look like? It's obviously a myeloproliferative disease involving mast cells, probably misdiagnosed in many patients through presentation to allergists rather than hematologists. And really, current therapies are symptomatic at best. In the aggressive form of the disease, rapid progression over a 2 to 3 year period. In the indolent, it's mainly treated by allergists asymptomatically from mast cell burden. So there are rare cases of mastocytosis that can be treated, as I've said, with imatinib, and these are the 5% of patients who do not have this mutation, and these respond very well to imatinib, and this really validates the target is the KIT target in this particular disease. There have been clinical trials with a very broad spectrum styroscorin analog known as PKC4-2. This has shown good results in phase 2 studies. However, this is a very dirty drug and with very poor pharmaceutical properties, so it's unlikely that this will make it through onto the market. So this is the compound we are currently progressing in development. This is the selectivity at 3 micromolar. It's a very potent inhibitor, so even in cell culture at 10 micromolar, it almost completely blocks the mutant D816 mutation. Most of these other kinases are not modulated at these low concentrations. So it's a very selective inhibitor. This is what it looks like in a model of mastocytosis in animal. This is using a mastocytoma, a very aggressive mastocytoma. Shown on here is animals treated with vehicle. Shown here is dasatinib. Now, dasatinib is able to block this mutation, but not at clinically relevant doses. So shown here is the effect of dasatinib inhibiting tumor growth, and this is our development compound. However, with dasatinib, you get rapid body loss of body weight due to its toxicity at these doses, whereas with the blueprint molecule, we have relatively healthy animals. So as I said, this is progressing towards through development. We plan to start our phase 1 studies at the beginning of next year, and hopefully this will have a similar impact on this aggressive systemic mastocytosis as we saw in the Gleevec trials with imatinib. So I'd just like to finish off by thanking and acknowledging my collaborators. So for the work on imatinib, especially Jörg Zimmermann, who was responsible for the chemistry, and Elizabeth Buchtunger, my collaborator in biology. Alex Matter, who was the head of the oncology division, who supported this project through very difficult times, and was essential in allowing us to get this into clinic. And then some of my clinical collaborators, Brian Drucker, who I've collaborated with and continue to collaborate with since the mid-1980s, Charles Sawyers and George Dimitri, who did the GIST work. And then my colleagues at Blueprint Medicine, Christoph Langar, a pioneer in the genomic space from the Vogelstein lab, and some of the same faces that I've mentioned that were involved in the Gleevec development. along with David Schenkheim, who is responsible for a number of the development programs at Genentech. So I'd like to again thank the organizers for giving me the opportunity to address you, and thank you very much.